show us how individual genes or group of genes are involved in health and disease. Understanding genetic factors and disorders is important in learning about promoting health and preventing disease. But there's a question. Is there more to this discussion? And that is what our next topic is. What controls you? Is it your genes or is it your shit? This is a very interesting topic and a great speaker, Professor Aziz Rehman. He's Professor of Medicine at Rashid Latif Medical College, formerly Professor of Medicine at Services Institute of Medical Sciences, Senior Vice President, PSIM, Editor, Journal of PSIM, and former Editor-in-Chief of Escalapio, which was the official journal of services of Institute of Medical Sciences. His main areas of interest include diabetes, hypertension, and chronic hepatitis. He is known to all to simplify things and teach in the most easiest manner possible. So, sir, please take over. We are looking forward to this talk.
All right, I think, first of all, let me apologize for this name of my title. I thought it would be catchy and it will make people a little curious what is this about. You know, our fecal matter, half of it is almost bacteria. And bacteria are a microbiota. So when they say shit, actually it refers to microbiota of our gut. Disclosure, nothing to declare. And first of all, let me talk about the role of microbiota in health, and then we will very briefly, this is actually extremely vast subject. Even if I go through it quickly, I will be only touching just the important parts. You know, it is generally thought that fetus in the womb is without any bacteria. This is what we used to think, but now the evidence says even in the amniotic fluid, there may be some parts of bacteria present, not in the live form, but there may be some kind of microbiota there also. But this is how it develops when the child is exposed. First of all, depends how the child was born. Was it vaginal delivery or was it cesarean section? Then exposure to external environment, which is inherently filthy. Infancy and childhood, different microbiota develops in different parts of the body, and then they become stabilized. Almost at the age of 3 years, everybody has a unique microbiota, and then it does not usually change, but during acute illnesses, it may be perturbed slightly, but then it is stabilized again. There's so many factors which actually contribute what exact microbiota you would have. So it is the exposure to the external, potentially filthy environment which helps our microbiota to develop. So I took this slogan from an advertisement, this is not conflict of interest, but this is just to highlight that dirt is indeed good to develop our immunity and our microbiota. Now, every part of the body has different microbiota, particularly gut. Gut is very, very rich in bacteria and many other organisms, but all body surfaces, all body orifices, they have unique bacteria and other microorganisms. Now, yes, okay. So this is how in the first 2 or 3 years microbiota develops, and then it remains stable and remains stable throughout our life. But during acute illnesses, there may be slight disturbance, but then it becomes, in most cases, it becomes stabilized again and healthy microflora is restored. In some cases, of course, this disturbance becomes permanent and patient develops a disease. Now, it is actually an essential organ. Maybe if I say life would not be possible without microbiota, I don't think I'm exaggerating. Now, let's give you some facts. 13 trillion human cells, and we have 130 trillion bacterial cells. So 10 times. You have 13 trillion your own cells and inside 130 trillion bacterial cells, every normal healthy person. Almost 1,000 bacterial species and many other organisms. 20,000 human genes and 2 million bacterial genes, 10 times. And if we isolate the total DNA from your body, 99% of that DNA would be bacterial. Only 1% comes from your host cells. So these are the staggering figures. So if we measure our total microbiota, the weight is equivalent to our brain. So I think it is plausible that this rich microbiota in our body has some essential role. Of course, we will examine it, what kind of role it has. Now, microbiome or a second human genome, we all know our genes control. My height, my complexion, my hairstyle, my way of eating and my way of talking is all determined by genes. If you examine your genome, 99% homology. Somebody sitting in this hall is 99.5% similar to another person who may be sitting in Chile or New Zealand. Only 0.5% diversity as far as genome is concerned. But when we examine microbiome, only 5% homology. Every person, the person sitting to you, the next to you, has got 95% different microbiota. So that is what makes you actually unique. Every individual has a unique signature microbiome like your DNA. And it is definitely possible that if the science is fully developed, it will be possible to recognize somebody from the microbiota. Now, if I compare a very simple thing like plant of rice, it has got 40,000 genes. And in human, there are only just over 20,000 genes. But there are additional 2 million microbial genes. So I believe these genes, they work together. And that is how this complex body functions are performed both in health and disease. So based on this new concept, International Human Microbiome Consortium was developed in 2007. They had series of meetings and they have developed this new science. And you know, conventionally, microorganisms are studied under microscope and their stain and their culture. Do you know that less than 20% of the bacteria are culturable? Other microbiota, 80%, you cannot even culture them. You cannot even stain them and see them. They're hidden. But thanks to this new technology based on 16S rRNA gene sequencing, now apparently there may be 2 E. coli which are exactly similar looking, same staining characteristic, same morphological characteristic, but based on this 16S RNA, there may be millions of subtypes of E. coli. So this is how we have gone into new era. There are simulators of the human intestinal microbial ecosystem. They have developed an artificial environment which is very much like our colon, because colon is the host of microorganism, and that has helped to study this new organism. iChip, iTip, these are the applications which can be used to study these organisms. Now, how host, that is ourself, which is only 10% if we count the cells. If we respect democracy, which we have to, 10% of our cells are our own host cells and microbiota 90%. how do they interact? Now you can see the human cells and microbiota, they interact either way. Both affect each other and both are affected by the environment. And this is the complex interplay. Now let me now give you some idea how we, the host, affect our microbiota. We are not yet talking about disease, we are just talking about their normal role. So how this gut microbiota, how we affect them. For example, mode of birth. I already mentioned if there is vaginal delivery, the likely microbiota is of different type, and cesarean section, microbiota will be of different type. And that may actually translate into future diseases. Diet, whether the baby was breastfed or formula fed, whether in the childhood and in the adult life was the diet primarily plant-based or meat-based, that can determine your microbiota. Usage of antibiotics. And we are so fond of prescribing antibiotics, people don't even need a prescription. But let me make a point, that 90% of the antibiotics are used in agriculture industry. So even if you do not take any antibiotic, you are still taking indirectly through the vegetables and fruits, and there is effect. And so antibiotic, they definitely have effect microbiota. Then drugs, any drugs like proton pump inhibitors, metformin, statins, laxatives, many other drugs are known to affect our microbiota. Diseases, inflammatory bowel disease and irritable bowel syndrome. In both, microbiota are of different type, but it is hard to tell at this stage which is the cause and which is the effect. And sleep, even our sleep pattern, our this circadian rhythm can affect our microbiota. And pets, you may be surprised to know that exposure to pets in early life actually prevents many diseases. Exposure to pets, in our culture, it is generally considered not right thing to be exposed to pets or animals in the childhood, but it actually helps. Now the second part, how our microbiota affects us. It affects us in digestion. There are several things which our intestine, our pancreas cannot digest. We need the help of a microbiota, especially the cellulose, the plant origin. And many bacteria, they actually produce folates, vitamin K, biotins, riboflavin, cobalamin. And then their defense, definitely they have a very, very important role in defending against colonization by opportunistic pathogens. And they also help in the development of our immunity. Very, very important. And then, 5 to 10% of the daily caloric needs are met by the calories which are harvested by our microbiota from those nutrients which would otherwise go waste. So 5 to 10% of the calories, they come from the microbiota, in other words. And they produce several short chain fatty acids such as acetic acid, propionic acid, and butyric acid. They all have some very healthy roles in our body. And then there's concept bacterial translocation. When bacteria get entry to the blood, they can cause bacteremia, endotoxemia. That is, of course, a pathogenic state, but by bacterial translocation, I means that some components of bacteria, they actually get translocated into the circulation, and they have effect on the liver and fats and other metabolism. So this is how our microbiota affects us. Now, microbiota and human disease. When I search this topic, guess, this is the PubMed. I'm sure everyone is familiar. This is the search engine which gives you the medical literature, which are published in the peer-reviewed journal. 19,940 papers on this topic, gut microbiota and human disease. And there has been a lot of interest in the last decade or so. So a lot of interest in this field. Now this is, there are theories about many diseases that they could be linked with our microbiota, but this is a common ground theory. Now you can see interaction of environment and host and microbiome, environment versus host, host versus microbiome, and this is very nice relationship, truly win-win situation. We live in harmony, we benefit from microbiota, microbiota is benefiting from us, but something goes wrong and this relationship is disturbed. And this disturbed relationship is called dysbiosis. Now, this dysbiosis can further cause a disease. But it is difficult to tell at this stage, is it the dysbiosis which has caused disease, or is it the disease which has caused dysbiosis? It could be either way. But very important concept I'm going to explain to you. If you have this animal, and if you transfer this dysbiotic microbiota to a healthy animal, and if that healthy animal develops a disease, that will be proof of concept that this microbiota is actually responsible for that trait or that disease. And this has been seen, and this has been shown in this Clostridium difficile infection, autism spectrum disorder, asthma, obesity, at least in lower animals, but some cases in humans also. So this is the common ground theory, how a dysbiotic organism can cause disease. Now, when we examine the role of microbiota in different diseases, these are 5 areas of interest. There are actually more. I will just briefly examine the possible role of microbiota in these 5 groups of diseases: infection, visceral diseases like gut, liver, and brain, allergic disorders and autoimmune diseases, metabolic syndrome, and obesity, which is perhaps the biggest disease of today, and atherosclerosis, which is the ultimate killer. So there is potential role of microbiota in the pathogenesis of these diseases, and there is at least theoretical possibility, but by modulating, we may be able to prevent these diseases. Let's first take infection. And all of you know this Clostridium difficile infection. So the theory is that somebody developed some infection which was to be otherwise self-limiting, but that person was given antibiotic, and that led to dysbiosis. Mostly, even after an antibiotic, dysbiosis will be reversible, but in some unfortunate person, that would promote the growth of Clostridium difficile infection, and that would lead to very severe colitis, which would be life-threatening. Not only that, it may lead on to develop what is called multi-organ dysfunctional syndrome. So it becomes a systemic disease. Now, can it be reversed? Of course, it can be reversed. Now, we have seen in those patients who have this Clostridium difficile infection, which are refractory to the most potent antibiotic, if they are treated with this FMT, fecal microbiota transplantation, there are various techniques, there is 94% response rate. So very, very definitive proof that transplantation of fecal materia can help in the treatment of Clostridium difficile. But there is a possible downside also. There is transmission of infection and unwanted trait. What I mean by that, I will show you by an example. And that is why we will have to select the donor. Not that anybody can donate fecal matter, but we will have to choose donors also. Now, infection with H. pylori, you know H. pylori infection is extremely common, and it may cause dysbiosis, then APD, and then it is definitely possible that it may lead on to develop cancer. There are many other infections. I might not be able to go into details, but bacterial vaginosis may increase the chances of preterm birth. Some woman who had some specific bacterial vaginosis, of course the symptom vaginosis will be there, but symptom may be silent, but this infection through this microbiota can actually cause preterm birth, acquisition of sexually transmitted infection. Of course, there has to be sex, and there has to be the partner has to be infected, but if this person has got infection due to some specific microorganism, that will promote the acquisition of these infections and poor responses from antiviral drugs. So this is about infection. Now let's talk about the visceral diseases, gut, liver, and brain. I'm quickly going through because the time would not allow me to go into the detail. So let's first take gut, liver, and brain. And you know there is possible link between gut microbiota and gastric cancer, possible link between gut microbiota and colorectal cancer, Barrett's esophagus and esophageal carcinoma, Crohn's disease, and ulcerative colitis. All these diseases have been linked with gut microbiota. I have already given a disclaimer. It is not possible at this stage to tell which is the cause and which is the effect, but there is definitely correlation. And there is a hypothesis. H. pylori, has been classified as class 1 carcinogen by WHO. And the theory is that it causes atrophy and metaplasia and dysplasia and then finally cancer. And these are the bacteria called enterotoxigenic Bacteroides fragilis. They produce a protein called fragilicin, and that reduces the production of helpful short-chain fatty acids. This is how gut microbiota are related to these some gastrointestinal infections and cancers. Now let's examine the association of microbiota and liver, the so-called gut-liver axis. If you have a predisposing condition, a specific gut microbiota can predispose to the development of spontaneous bacterial peritonitis, portosystemic encephalopathy, hepatocellular carcinoma, autoimmune hepatitis, and non-alcoholic fatty liver disease. Of course, you have to have their own causes, but presence of a specific type of microbiota may predict that the chance of developing these conditions is more. Dysbiosis may lead to increased growth of Enterobacteriaceae, which is harmful, and the reduced production of Bifidobacterium, which is relatively friendly. And it may increase the production of ethanol, ammonia, and acetaldehyde. Bacterial translocation may lead endotoxemia. This is not the usual sense of endotoxemia. This means some bacterial products are circulating which have metabolic and other effects, and this may cause immune dysfunction. Now the gut-brain axis, our eating behaviors, how much we eat and also our eating preferences, they are linked with gut microbiota. Our mood is related with gut microbiota. Memory, Alzheimer's disease, major depressive disorders, autism disorder, multiple sclerosis, all these brain disorders have been linked with specific type of microbiota. And these specific microbiota means based on those RNA, 16S RNA. Dysbiosis may lead to production of serotonin, and serotonin through vagus nerve actually have effect on the brain. So when we say my gut feelings, we may be actually talking science. You all know an old man coming and saying that I have problem, gas here, and then it goes to the brain. We always laugh on this. But there may be actually science in that. And our gut microbiota may be actually affecting our mood and our memory, and it also predisposing to certain diseases. So third is allergic disorders and autoimmune diseases. Very, very strong relation with microbiota and autoimmune diseases and allergic disorder. Asthma, for example, allergies, and type 1 diabetes, because type 1 diabetes is also primarily an autoimmune disease. So there is link between gut microbiota and these diseases. This is an organism, Clostridium sensu stricto. It predicts allergic disorders because it tends to increase the production of interleukin 10 and reduces interleukin 4 and 5 and 13. I don't know how this thing works, but this is the proposed mechanism. Bacterial genes, they increase production of something called 12-13 dihome, and exposure to pets and cockroaches, they reduce the incidence of future asthma. And Lactobacillus johnsoni reduces asthma, and early life antibiotic or hyperhygienic condition. This might actually be difficult to believe. Hyperhygienic condition, the superhygienic state, the directly proportional to the incidence of type 1 diabetes and autoimmune disorders and allergic disorders. Like in Scandinavia, type 1 diabetes is up to 30%. In our country, it's just 5%. So I think they could learn from us how to reduce type 1 by producing filthy environment. Now the metabolic syndrome, as I said, the biggest disease of today, and obesity. The things are even more interesting. Gut microbiome is related to the weight we have and the metabolic syndrome and type 2 diabetes. Type 2 diabetes is undoubtedly one of the biggest challenges, and this is affected by the diet and the environment. And in certain patients, firmicutes are directly proportional to the BMI, specific type of firmicutes. And Christensenella is indirectly proportional to the BMI. And guess, this is the very interesting fact, the predictive value is 90%. If you have a specific type of firmicutes or Christensenella, your weight or BMI can be predicted accurately by 90%. So there is that strong relationship. And gut microbiota and insulin resistance, somebody with different gut microbiota would show different glycemic response to the same food, irrespective of their otherwise metabolic health. So because of the gut microbiota, the response to glucose may be different. And certain microbiome's signature predicts type 2 diabetes. Now, the hypothesis behind this is very complex picture. This is taken from, I think, New England Journal of Medicine. And I have made a simplified version. There is a specific type of Escherichia coli which produces a protein called CLPB. Now, this protein has got the effect very similar to GLP-1 and peptide YY. And you know these peptide YY and GLP-1 both are anorexogenic hormones, and they tend to reduce your food intake and you tend to have less weight. And if this organism is not there, then you are likely to develop problems. So this is going to be a friendly organism. It reduces insulin resistance, it reduces metabolic syndrome, and it reduces the chance of diabetes if you are lucky to have this guy which produces CLPB. Now, association of microbiota with obesity, very, very interesting observation I'm going to share. If there is an animal who is treated with microbiota taken from an obese person, that animal may put on weight. And this has been shown, definitely. But we would be more interested if the reverse is true, because all of us are very, very obsessed to lose weight. So if the reverse experiment is successful, then that would be very, very exciting. So I think if the microbiota of 10 people may be in high demand, and they may become filthy rich. I'm sure nobody wants to gain weight, maybe 5%, but everybody else wants to lose weight. So if science develops, then I think this microbiota may be really helpful. And this also explains why some people just can't lose weight despite dieting. Everybody says that I don't eat much. But I have gone through patients' diets, and they tell me half a toast in the morning, half a bread in the afternoon, and then I sometimes feel that this is not even enough, but they are overweight. So it is maybe related to gut microbiota. You know, I have already shown you they produce about 5 to 10% calories from fiber, and there is a theoretical possibility that our microbiota can produce energy from air and water also. So if somebody is like this, he is not to be accused for being fat, and if you're like this one, you cannot be given credit for being slim, because it may be your microbiota which is determining your physique. Now, there was an interesting breaking news. The slimy bacterium may fight obesity. This is a bacterium called Akkermansia muciphilia, and you can see in those who have this, insulin sensitivity is high. That means resistance low, total cholesterol is low, and gamma GT, which is a marker of fatty liver, that is low, and WBC count, which is also a risk factor for coronary artery disease. There are some interesting papers, insight into the role of gut microbiota in obesity, pathogenesis, mechanism, and therapeutic perspectives. And this is a very, very interesting case report I'm going to share with you, weight gain after fecal microbiota transplantation. There was a woman who had Clostridium difficile infection, and all antibiotics were tried, none worked, and she was 61 kg. She was offered this FMT, and she chose her daughter, who was healthy, and she had just 77 kg of weight. And the woman with ICD, she recovered, but continued to gain weight. She did not regain her lost weight, but she continued to gain weight, and she achieved 80 kgs from 61 kg. And it was because of the donor microbiota was those which were responsible for this weight gain. So this can be actually used therapeutically also. So now the last one, atherosclerosis, undoubtedly very, very important disease. So if we can find a solution of atherosclerosis and microbiota, that would be really great. Now, microbiota, Escherichia coli, this is something which comes from the diet, phosphatidylcholine and L-carnitine. Now this gets converted into trimethylamine N-oxide, TMAO. This one, along with atherogenic diet, may predispose to atherosclerosis. Now, if we intervene this by giving a dietary supplement which can stop the conversion and production of TMAO despite atherogenic diet, these experimental animals do not develop atherosclerosis. So this is really great. By modulating these organisms over diet or working through them, I think we can prevent metabolic syndrome, obesity, and atherosclerosis. Now, gut microbiota potential therapies, stool transplantation is now proven therapy for Clostridium difficile infection, and this one can work for atherosclerosis if the theory proves correct, and Bacteroides fragilis can work for our anxiety and depression, Lactobacillus, IBD, CLPB producing Escherichia coli, adiposity, metabolic syndrome, and diabetes, and bacteria, many bacteria, they produce specific antibiotics. They may provide future antibiotics, and early life probiotics may be used to prevent type 1 diabetes and asthma. And all these actually will probably develop to probiotics and prebiotics, because it would be hard to ingest or to take FMT. Although there are ways, like for example, one can take capsule, or somebody can be given through endoscope or through the colonoscope. So summary, everybody has a unique signature microbiota. You are truly unique by your microbiota more than by your genome. It undoubtedly plays a crucial role in our health and disease. Perhaps our life would not be possible without them. So next time you call somebody a piece of shit, you might actually be giving him compliments, because shit is capable of doing so much good things. The future might see new lab tests, new therapies based on microbiome, and we might have to rewrite the pathogenesis of many diseases. And in this new science, how much is the hope and how much is the hype is yet to be known. What controls you, is coming back to my question, is it your genes or is it your shit? I think it's a teamwork. Both are indispensable. But if you go by numbers, so 99% of your DNA material comes from bacteria and only 1% from your host cells. So what we see is just the tip of iceberg. We still have to go a long way to uncover microbial mysteries. And we know that good diet, which is based on vegetables and yogurt, that actually gives you a healthy microbiota. These are the references. I dedicate this lecture to my friend, my mentor, Professor Faisal Masood. It is because of him that I got interested in this topic.